Hello, this is Miss Dagenford, and we are going to continue our discussion on cell signaling by looking at second messengers. Now, this got a lot of confusion I heard today, so we want to try to clear that up. Second messengers are really no more than the guy that's behind the door. So if the ligand is the first messenger, and they're knocking on the door, and here's our cell membrane. This second messenger is the one that's going to wake up, hear the message, and then send it on to that signaling cascade. So they're not, this diagram can be overwhelming at first, but notice there's something familiar over here. Here's our ATP, and ATP can be converted through an enzyme. So here's our enzyme, there's our ACE, through adenylyl cyclase. That's going to break down ATP. It's going to strip off two phosphates, and then we're going to have cyclic ANP right here. Now, you can then remove an oxygen and you can have AMP, but that's not going to work. We want that cyclic ANP. Now, here's a good example of how things can go wrong. Here's our case study, and this is Vibrio cholera. This is a bacterial disease, and of course it causes, just as the name implies, cholera. So what this is our large intestine. Some of you from anatomy class will recognize this. Here's our large intestine, ascending, transverse, descending. And if I were to blow up the lining here, we'd have our little cells. And if we were to make one bigger, you'd have your G protein coupled receptor, your G protein, and what will happen is you've got the cholera bacteria that's growing on top of the lining of the small large intestine, and it's making quite a lot of toxins. And remember, here's our quorum sensing. It's going to make these toxins, and these toxins are going to be affecting that G protein such that it cannot break GTP down. So it's stuck on GTP, which means it is always on. Now, what happens is once that G protein is on, it's going to go to our adenylyl cyclase and convert the ATP down to cyclic AMP. And now we're increasing cyclic AMP. That in turn, that concentration keeps rising. That will affect a sodium pump, and what will then happen, this gets turned on, you start pumping sodium out, which unfortunately for those who are inflicted with cholera is also going to cause water. Good practice with our osmolarity, it's also going to cause water to flood back into the intestine, and then we have copious amounts of diarrhea, which can be, if not controlled, quite disastrous. It can be fatal. So you can see that if we affect, here's an example of a bacterial toxin affecting the ability of that GTP to switch off, and it keeps it on, and then we build more and more and more cyclic ANP. That affects the sodium transporters. So quite important molecule, that cyclic AMP. Now, calcium is another second messenger, and what a lot of cells can do is it can store calcium in the endoplasmic reticulum. Some of you actually, if you think about it, we talked about this in muscle cells in the specialized sarcoplasmic reticulum, and that stored calcium, and so calcium becomes a really important messenger. For instance, here would be the endoplasmic reticulum. Here's our calcium being stored in here. And the calcium can then signal important things to happen, like the muscle contracting or the synaptic vesicles signaling. So calcium becomes quite important as well. There's another one that happens internally to the cell, and they're called inositol triphosphates. and diacylglycerol, subsequently abbreviated IP3s and DAG. And so if you have a ligand, these molecules can break free of or move around on the membrane, and then they can send signals as well throughout the cell. So all of these end up being really important because they're kind of the first inside the cell. That's what makes them the second messenger. The ligand would be. Here's a good example of our IP3 and our diacylglycerol. They're membrane bound, but then they can break off, and then they're sending signals. Now, this happens to be within the cell. Here's our endoplasmic reticulum that's then sending out the calcium, and then the calcium is going to be getting our cell messengers. So in this example, the calcium and these molecules, the IP3, they're considered both second messengers. And I think what you have is the development of what we know over the years. We realize that they're both involved, even though one's second and one's third. We both call them second messengers. I know that can be frustrating. Now, we get to responses, and here we can actually go pretty fast. We've got the phosphorylation cascade, which, remember, is amplifying the signal. So we've got one ligand, and then we have our cell membrane. We've got our receptors on the G proteins or the tyrosine kinase phosphorylation. They're going to be sending off signals, and here we have one molecule that can activate 10 molecules. And I probably didn't get 10 there. And then each of these can signal 10. And then within 4 rounds, we've got thousands or 10,000s, if we use that math, of all of these molecules that are going to be sending signals to do something, and we can increase the response quite high. And then here it goes into the nucleus, and in this case is acting as a transcription factor, and it's going to turn on a transcription factor, which is going to cause the RNA polymerase to bind, and boom, we're going to get our mRNA, and then we're going to get a protein, yay. And so that's our response. But it can also turn on other proteins, or it can cause the vesicles to bond to the membrane like we saw with insulin. So those are a lot of responses, and we don't need to know each and every one of them, but we'll go over some examples tomorrow in class. So that gives you kind of the end result. All of these molecules basically are going to change something on the inside of the cell. Increase cell division, or decrease or stop cell division, or increase transcription or turn off transcription, cause something to change within the cell. And we're going to look at how that happens in the eye tomorrow. Now, what we'll look at, here's our amplification, by the way, and you can see here's our numbers, 10 to the second, 10 to the second, 10 to the fourth, 10 to the fourth. That increases the amplification. So we'll continue and look at some of the examples that happen, like all of the different variations of how these things can interact, and then we'll start looking at the nervous system tomorrow as well. So I hope that gives you kind of a conclusion to what we're looking at. Have fun and see you tomorrow.